In the summer of 2013, divers off the coast of Washington state began noticing something wrong with the sea stars. The sunflower sea star, Pycnopodia helianthoides, is the largest sea star in the world. It grows to the size of a bicycle tire, spreads up to 24 arms across a meter of seafloor, and moves through shallow coastal water at a speed that is, for a starfish, genuinely surprising. It is among the most formidable predators in the kelp forest ecosystem, consuming sea urchins, clams, snails, and other sea stars, with an efficiency that keeps the populations of everything below it in check. On a healthy Pacific reef in 2012, you could find sunflower stars in dense aggregations across the rocky bottom, dozens in a single dive site, arms spread wide, moving purposefully through the kelp. By late 2013, the divers were reporting something different. Sea stars, not just sunflower stars, but ochre stars, giant pink stars, rainbow stars, mottled stars, were developing white lesions on their upper surfaces. The lesions expanded. The tissue around them softened. The arms began to twist and curl. Then, over the course of a few days, the arms separated from the body. The remaining central disc dissolved into a soft white substance and dispersed into the water. The animals were, in the most literal sense, coming apart. The disease spread north to Alaska and south to Mexico. It moved through population after population. By the time the initial outbreak slowed, researchers had documented the largest marine invertebrate mass mortality event ever recorded. More than 20 species of sea stars were affected. The sunflower sea star, which had previously ranged along the entire Pacific coast of North America, in numbers that made it one of the most common subtidal animals in the region, lost more than 90% of its global population. In California, subtidal surveys that had recorded hundreds of sunflower stars per dive site recorded 0. The species was added to the International Union for Conservation of Nature Red List as critically endangered. 5 billion sea stars by the most comprehensive estimates. Gone. And for 12 years, no one knew why. The search for the cause of sea star wasting disease became one of the most actively investigated mysteries in marine biology. The symptoms pointed toward a pathogen, something infectious given the speed at which the disease spread through populations and the consistency of the clinical presentation across dozens of species and thousands of kilometers of coastline. The pattern of spread, moving roughly along the coast from north to south, suggested transmission through water rather than direct contact. The fact that animals in aquariums, supplied with water pumped in from the ocean, developed the disease and died, while animals in closed systems with filtered water remained healthy, provided early evidence that the causative agent was waterborne. Initial research in 2014 identified a candidate, a densovirus. A type of small, single-stranded DNA virus, appeared to be associated with the disease in sequencing studies of affected tissue. The finding was published, received significant attention, and shaped the research direction for the following years. Subsequent work, however, produced contradictory results. The virus was found in apparently healthy animals. Animals experimentally exposed to the virus did not consistently develop the disease. Populations in some regions showed signs of recovery without any change in viral exposure. The viral hypothesis, while not definitively ruled out, became increasingly contested. The mystery deepened. Warmer water temperatures appeared to correlate with faster disease progression and higher rates of affected animals. The extreme marine heat wave that affected the northeastern Pacific beginning in 2014, the period researchers called the Blob, coincided with some of the most severe outbreaks. But temperature alone did not explain the pattern. The disease had appeared before warmer conditions arrived, and it persisted in cold water, where the temperature connection should have been weakest. Other hypotheses emerged: bacterial involvement, multiple interacting factors, a stress-related immune suppression that made animals vulnerable to pathogens they would normally resist. The scientific literature accumulated dozens of papers, each adding a piece of a picture that refused to resolve into a coherent explanation. The animals continued to deteriorate in affected areas. The sunflower star remained critically endangered. The cause remained unknown. On August 4, 2025, a paper published in Nature Ecology and Evolution announced the result of a multinational, multi-institutional research effort that had been running for years. The causative agent of sea-star wasting disease in sunflower stars is a strain of the bacterium Vibrio pectinitida, specifically a novel strain designated FHCF3. Isolated from the coalomic fluid of a diseased sunflower star at the Hakai Institute in British Columbia. The team had applied Koch's postulates, the classical framework for establishing that a specific pathogen causes a specific disease, with rigorous experimental design. They exposed healthy sunflower stars to tissue extracts, coalomic fluid, and water from wasting animals. The exposed animals developed the disease. Control animals that were not exposed did not. They isolated Vibrio pectinitida strain FHCF3 from the coalomic fluid of diseased animals. They cultured the bacterium, exposed healthy animals to it directly, and confirmed that the bacterium alone was sufficient to cause disease and subsequent decline in exposed animals. They sequenced samples from both experimental and field outbreaks and found the bacterium present in high proportions in diseased individuals. The cause of the largest marine invertebrate epidemic ever recorded had been a strain of Vibrio bacterium, the same genus responsible for cholera in humans, for widespread coral disease, and for mass mortalities in shellfish aquaculture worldwide. The bacterium had been present in ocean water, had entered sea stars through their water vascular system or through compromised surfaces, and had spread through populations in a pattern consistent with waterborne transmission. Why 2013? Why that scale, that speed, that geographic reach? The paper does not fully resolve this question. The role of warmer ocean temperatures, which are known to increase the virulence of Vibrio bacteria and to stress host immune systems, likely contributed to the severity of the outbreak. Whether prior outbreaks of sea star wasting disease, documented in smaller events during the 1970s, 1980s, and 1990s, involved the same bacterial strain or different pathogens is not yet established. The mechanism by which the bacterium causes the specific disintegration pattern, the tissue softening, the arm separation, the dissolution, is the subject of ongoing research. But the central mystery, the question that had occupied marine biologists for 12 years, was answered. The discovery was described by researchers in the field as the discovery of the decade in marine disease science. Understanding the cause matters enormously. But what happened after the sea stars disappeared matters in a different and more immediately visible way. The sunflower sea star is a keystone species. The concept of a keystone species, an animal whose presence or absence has an outsized effect on its ecosystem relative to its own abundance, was in fact largely developed from research on sea stars. In the 1960s, ecologist Robert Payne conducted experiments on rocky Pacific coastlines in which he physically removed a sea star species, Pysasta ocraceus, the ochre star, from intertidal plots and observed what happened. What happened was the near-complete ecological restructuring of the plots. Mussels, which the sea star had previously kept in check, expanded to cover the rock surface. The diversity of the intertidal community collapsed from more than a dozen species to essentially one. The removal of a single predator species had cascaded through the entire community. Payne named the animal he had been studying a keystone species, after the central stone of an arch, which, if removed, brings the whole structure down. The removal of sunflower sea stars from Pacific subtidal ecosystems beginning in 2013 produced exactly this kind of cascade, at a scale that Payne's experimental removals had hinted at, but never approached. Sunflower stars are voracious consumers of sea urchins. Sea urchins are voracious consumers of kelp. In a healthy kelp forest ecosystem, sunflower stars keep urchin populations at levels that allow kelp to grow and maintain the structurally complex canopy that dozens of other species depend on for habitat and food. When the sunflower stars were removed, suddenly across hundreds of kilometers of coastline simultaneously, the urchins had no significant predator. Their populations expanded. In some areas, urchin densities increased fourfold within months. The urchins moved into kelp forests that had previously been healthy and began grazing at a rate the kelp could not sustain. Kelp forests that had persisted for decades disappeared within seasons. In Northern California and Oregon, where the sea star loss was combined with warm water anomalies that further stressed the kelp, bull kelp forests collapsed across more than 90% of their historical range in some regions. The transition was rapid enough to be captured in satellite imagery. Dense green canopy visible in 2012, open barren urchin-dominated rock by 2015 and 2016. The ecosystem that replaced the kelp forest, dominated by sea urchins with minimal kelp, lower fish diversity, reduced habitat complexity, is sometimes called an urchin barren. It is stable in its own way, which is the problem. Urchin barrens are self-reinforcing. The urchins prevent kelp from re-establishing by grazing on kelp recruits before they can grow. Without established kelp providing cover and food for other species, the food web that would otherwise support sea otter populations, fish communities, and the full range of nearshore biodiversity is not there. The system is stuck. Recovery of sea star populations has been slow and uneven. Ochre stars, more resistant to the disease than sunflower stars, began showing partial recovery in some regions within a few years of the initial outbreak. Sunflower stars, which were more severely affected, have shown minimal recovery across most of their range. Individual animals have been spotted. In 2023 and 2024, researchers and citizen scientists reported small numbers of sunflower stars at locations where none had been seen since 2014, raising cautious optimism. But population surveys have not shown meaningful recovery at scale. The identification of Vibrio pectinicida strain FHCF3 as the causative agent opens pathways that were not previously available. Knowing the pathogen makes it possible to develop screening tools to detect its presence in water and in animal populations, providing early warning of outbreak conditions before widespread impact. It makes it possible to test whether some individual sea stars carry resistance factors that could be selected for in captive breeding programs. It makes targeted intervention strategies, probiotics, vaccination, habitat management, conceptually feasible in a way they were not when the cause was unknown. Whether these strategies can be implemented at the scale of the Pacific coast, and whether recovering sunflower star populations could re-establish in regions where urchin barrens have now been in place for more than a decade, are open questions. The trophic cascade that the sea star loss triggered is not automatically reversed by the presence of more sea stars, any more than removing a dam immediately restores a river ecosystem. The urchins, the kelp, the fish communities, the predators above them all have to readjust, and the timescale for that readjustment is measured in years to decades, not in seasons. The story of sea star wasting disease is not primarily a story about sea stars. It is a story about connection. The sunflower sea star was the central link in a chain that ran from the sea urchins it consumed to the kelp that the urchins would otherwise overrun, to the fish that sheltered in the kelp canopy, to the marine mammals that fed on those fish, to the intertidal communities that depended on the same rocky substrate, to the human communities whose livelihoods depended on the fisheries and ecosystems those reefs supported. When one link broke, the chain did not simply sag. It came apart in ways that are still playing out more than a decade later. Keystone species are defined by exactly this property. Their removal has effects that exceed what their own biomass and behavior would suggest. The concept, which began with Robert Payne removing sea stars from tide pools in the 1960s, is now illustrated at continental scale by what happens when sea stars are removed not by a researcher with a crowbar, but by a bacterium in the water. The cause is now known. The pathogen has been identified. The research that 12 years of effort finally produced gives the people working on recovery something concrete to work with. What 5 billion sea stars built, over millions of years of coevolution with the ecosystem they shaped, cannot be quickly rebuilt. But the understanding of what was lost and why is now at least complete enough to begin. If you want to see what happens to the deep ocean ecosystem when its own keystone species are removed, the version of this cascade that plays out 1,000 meters below where sunlight reaches, that video is here.